Okay, so now we're going to jump more into the physiology. And kind of like the cardiovascular system, there is a lot of physics in here, but don't let it scare you. It's really not that bad. So when we talk about respiration, it is important to realize this happens in a couple different phases, or people might mean different things by respiration. So notice, we're calling this entire process of getting oxygen in, moving it to the blood, moving it to the cells, and then CO2 all the way back out. We're calling all of that respiration, right? So this entire box. But we can then also talk about external respiration, right, which is this first half, and internal respiration, which occurs later. Internal respiration you could think of essentially being equivalent with cellular respiration, right? So we're going to get the oxygen into those cells. They're going to do the cellular respiration, break down the glucose in the presence of oxygen, and release CO2. So internal respiration, that's actually like cellular respiration, right? Okay, so we're going to spend more time talking about external respiration. We'll talk more about this part of the picture in chapter 25, right? When we get into cellular respiration more. So in external respiration, we're really talking about three different things. So the first one, so this is like external respiration A, right? Or one, is pulmonary ventilation. Okay, this is the actual movement of air in and out of the respiratory tract, right? We're trying to deliver it to and from the alveoli. Okay, we'll come back to that. The other parts of external respiration are this gas diffusion, right? So we see that we need to get gases across the respiratory membrane, right, between the alveoli and the capillaries. And so we want to move oxygen into the blood, obviously, and CO2 back out. And then the third and final portion of what we would call external respiration is oxygen and CO2 transport, right? So how do we actually carry these in the bloodstream? And it's a little more complicated maybe than we would think to begin with. Okay, so let's look at each of these a little bit more in depth. So pulmonary ventilation, right? This movement of air in and out of the lungs. This is a physical process. What you're going to find is just like in the blood and in diffusion and in osmosis, right? What governs the movement of air is that air wants to move from areas of high pressure to lower pressure, right? So you could even like do that with your hands, right? If you squeeze and you increase the pressure in that little pocket of air you have in there, it wants to squirt out, right? Because there's less pressure in the surrounding room. So air wants to move from high pressure down to lower pressures, right? That makes sense. So what we're going to see happen is that the lung utilizes this information, right? Basically, the lungs are going to cycle just above and just below atmospheric pressure. And so atmospheric pressure comes up a lot in here. And so kind of what you need to think, oh, that's terrible. What you need to think about is that as you're sitting here watching this video, this is you. Yep. Okay. There is a column of air on top of you, right? The atmosphere is sitting on your head. Now, the atmosphere is lightweight, right? You don't even have to hold it up, but the atmosphere is pushing down on you. It's resting on top of your head right now. Okay. So, and that pressure, we would measure this with a tool called a barometer. That pressure on average, we'll just use sea level for now, is 760 millimeters of mercury. So it's kind of a weird unit, but just like a thermometer would use mercury, these old barometers would too, right? So you could open one end and the pressure of the atmosphere down would push the mercury up on the other side. And this atmospheric pressure changes some with storms, right? High pressure tends to be clear skies. Low pressure, you could actually watch the mercury drop, right? And that would indicate that a storm was coming. Okay, but for us, we're just going to use Atmospheric pressure, this is the weight of the atmosphere sitting on your head. Sometimes you'll see people just use 1 atmosphere instead. 1 atmosphere is equal to 760 millimeters of mercury. So all we really have to do to get air to flow is to make sure for inspiration, right, what happens is all we have to do is increase the volume a little bit, right, the amount of space, and the pressure drops. Look how much room there is for all those gas molecules. Air would go rushing in. And that's exactly what we do. If we can make the pressure in the lungs 759 millimeters of mercury, air comes rushing in, right? And then exhalation, we're just going to make that container a little bit smaller, right? We decrease the volume. That pushes all these molecules closer together, and then they want to escape, right? So they're just going to flow. Now, what gets confusing here is they start giving you equations, right? So they give you the equation for Boyle's law, which is really just pressure is that's a 1, is inverse to volume. Now, in your book, I think they actually gave you K over V, and K is just a constant for temperature. We can ignore that. You can just stick with 1, right? Pressure, and the easiest way to say this is pressure is inversely, right? So 1 over something would be the inverse. Or think about it this way. As volume increases, pressure decreases, right? Those molecules are all spread out. As volume decreases, pressure increases. You've shoved them all together, right? So if you know that pressure and volume are inversely related, then you can use Boyle's law, right? You never have to calculate anything with this. Okay. So this is kind of, actually, I think your book had. So then when we want to change those pressures, what we end up doing is using a lot of the muscles of the thorax and sometimes a little bit of the abdomen as well. So in order to inhale, right, basically what we need to do is we need to increase the volume, because then pressure does what? Decreases, and air wants to flow in, right? So that's exactly what we're going to do here. So in order to increase the volume, we do things like, notice here the diaphragm contracting. The diaphragm is this big dome-shaped muscle, and so when it pulls down, that increases the volume of the lungs. You also can lift the rib cage, like they're showing here, right? And again, even in like quiet breathing, just sitting here watching, typically what's going to be lifting the rib cage are those external intercostal muscles. But again, that's going to kind of open up the thoracic cavity and increase the volume of the lungs. Now, other things that you need in order for this inhalation to work, right, is that you need the lung to somehow expand with, say, the rib cage or the diaphragm. And so what we're going to see is that the pleura is what helps us out here. So I'm switching back to a picture from my old book, right? Notice here we have the lung, and then this little white area here, now I'm coloring it in, that's the pleural space. So the lung tissue itself is covered by a visceral pleura, and then the thoracic cavity is covered by the parietal pleura, and the little space in between there has serous fluid. This is just like the pericardium, okay? But the serous fluid, so it decreases friction, they can move past each other, but they also then adhere to one another. So as the rib cage lifts and the parietal pleura goes with it, as long as there's serous fluid, that visceral pleura sticks, right? And so we open up the lungs as we open the rib cage. So I did want to point that out, right? You need the pleural fluid, you need that pleural space. So basically the surface of the lung is basically stuck to the chest wall, right? And the diaphragm. And so the lung tissue expands as well. Now, not only do you need the pleural space, the pleural fluid to do this, but the lung also needs to be compliant. So compliance refers to expandability. And this then deals with multiple different factors of the lung, right? We need the connective tissue to be expandable. We'll find certain diseases where it starts to stiffen up and no longer wants to kind of stretch. We talked in the anatomy video about surfactant production. You don't want all your little alveoli sticking shut or the lung becomes way less compliant, right? You can't get it to open. And so you need that surfactant production. And you also, and this picture I kind of like, right? It's showing how much the rib cage actually has to move. And so you also need the rib cage to be mobile. So there's certain diseases, even just like with aging, you might see a loss in that thoracic, the rib cage compliance, right? Or the lung compliance as that thoracic cage becomes less mobile. Okay. So then when we exhale, which we're showing over here, basically what happens is the diaphragm relaxes. So inhalation required actual muscular contraction. Exhalation is a relaxation of the diaphragm. It goes back to that dome shaped, and that is going to decrease the volume of the thoracic cavity and therefore the lung. Right? And we increase the pressure, and it only has to go to 761 if the pressure is slightly higher than atmospheric pressure. Air just goes rushing out. Okay? And so, again, this is what it would look like. This picture is even better, right? In pink here is the diaphragm. And then this blue is the pleural space. So the parietal pleura is stuck to the diaphragm. The visceral pleura is stuck to the lung. And as long as there's pleural serous fluid in there, they'll stick together, right? And so again, notice what happens here. They've just made this 0, right, 760. You only have to drop to 759 during inhalation to get air to come rushing in. And you only have to go up to 761 for air to go rushing out, right? Notice also the interpleural pressure. So in that kind of in-between space between the pleura, we get a little bit larger swings in pressure there. There's a real suction between those two layers. And then this final one is showing us the volume of air that we're able to move in and out of the lung. And so in a normal inhalation, we move about 500 mils of air into the lung or out of the lung, depending if you're talking about inspiration or expiration. So tidal volume is kind of that normal breath, right? What the volume is going to be. Okay, so hopefully this image helps a little bit. But again, it all comes back to that inverse relationship between pressure and volume. Okay, so then we have a bunch of different values here. So this is a result of these numbers. This readout is the result of something called spirometry. So you use a spirometer. You're basically breathing in a tube, right? And the machine is going to chart the changes in pressure, just like we were talking about. Or in this case, it would be the volumes of air that you're actually moving. So, and this is, well, let's just look through the one from your book. If you like the pictures from the other book, you do have the PowerPoint presentation of the module, and so you can look at that as well. So volumes that we're interested in here, absolutely tidal volume. So, tidal volume. Okay, tidal volume, we just mentioned, right, is about 500 mils, and this is the volume of air moved in and out in a normal breath. Okay, so about 500 milliliters. Now, if you're going along here and you're breathing in and out and in and out, normal breaths, right? And then you breathe in. Sorry, I can't do it. Breathe in, and then keep breathing in. Right into that maximum inspiration. This whole amount here is what's referred to as inspiratory reserve volume. Look at that, 3,000 milliliters. And of course, this varies a little bit person to person, but 3,000 milliliters, right? You have a way higher inspiratory reserve volume, or IRV, than you do tidal volume. Again, this is the amount that can be inhaled above and beyond what that normal breath is, right? 3,000 is a little more average for males. Females tend to be down by like 2,000, right? A lot of this has to do with just body size. Okay, so you have this inspiratory reserve. You could breathe in more if you really had to. All right, and so then we move back. We do a normal inhale, right? After our exhale, we do a normal inhale, and then we exhale. And then we find, oh, even after the end of that exhale, I can still breathe more and more and more, right? That maximum expiration, this is expiratory reserve volume. So again, the amount that can get exhaled after that quiet breath. Okay, they're listing about what 1,100 mils. There, other places I've seen more like 700. It's definitely smaller than inspiratory reserve capacity. Okay, also notice after that maximum expiration, there is still a lot of air in the lung here, right? Somewhere around 1,200 milliliters remains, and this is what's referred to as residual volume. So after the maximal exhalation, how much air is left. So this residual volume, keep in mind that basically you have that anatomical dead space. So you don't, how do I say this? You don't want the lungs to completely collapse, right? All the alveoli to close. So you have to leave some air in there. And so that's what we're going to see here in this residual volume. Okay, so those are the 4 important volumes. And then we move into this idea of capacities. And so capacities are going to link together a couple of these volumes. So for example, vital capacity. This is a super important one, right? So vital capacity is the maximum amount of air that you can move in a single respiratory cycle. So notice what it includes here, right? From a normal exhale all the way up, right? That would be your vital capacity. Okay. I'm sorry, all the way in and then all the way back out, right? So from here all the way in, your vital capacity. How much air can you move in a single respiratory cycle? If you just take the top half of that, like I was trying, the normal inhale and the maximum inspiration, that gives you inspiratory capacity. You can also do an expiratory capacity, exhale, and then continue to exhale. Total lung capacity, right, is different than vital capacity. Notice what total lung capacity adds on, right? It adds on that residual volume. Oh, one other capacity here. Let me get that out of the way. Right, this functional residual capacity. What we're looking at there, that is basically this expiratory reserve volume, right? So how much you can exhale after a normal breath, and then also that residual capacity. Those two values together are referred to as functional residual capacity. Okay, so these are important values to know. Again, this is another look at that, but it has the exact same thing. I do like this one writes horizontally. It's a little easier to read, and it does give you the abbreviations as well. But whatever you want. Okay, before we leave that thought too far, notice also, let me go back here. So the other thing though to keep in mind, right, is we can use these values then to calculate more like how much air we can move per minute. So you can take some of these values and then multiply them by the respiratory rate. So ones that you might see, let me get rid of all of that. Ones that you might see would be the respiratory minute volume. So as soon as you see minute, right, we're just adding, wow, I don't know why the pen keeps doing that. So respiratory minute volume, you'll often see abbreviated V sub E, a little dot on top of the V. And the dot is just telling you that it's per minute. Okay, so the volume here. So what you're going to do here to get this respiratory minute volume is you're going to take the tidal volume, the amount of air you normally move in a respiratory cycle, and you multiply it by the number of breaths, right? So you could do basically breathing rate I'll call it respiratory rate. My book abbreviates it F, right? But it's basically respiratory rate times tidal volume, T sub V. Okay, and so if tidal volume is 500 mils and if a normal respiratory rate is 12, how much air are you moving per minute? If the respiratory rate is 12 and the tidal volume is 500 mils, that should get you to 6 liters, right? And so that would be a very normal respiratory minute volume, 6 liters per minute. Okay, but one thing that we do have to keep in mind here is that part of each of these breaths, right? If you think about the tidal volume, you're blowing in this tube. When you inhale, there's going to be a portion of that. There's going to be a portion of that inhalation that never makes it to the alveoli, right? It's in that conducting space. It's in the trachea. It's in the bronchioles. And that's about 150 mils. I think they usually say like what your, what a healthy weight should be for you in mils. You can use 150. That'll work just fine. So they also set up a calculation to say, okay, well, what's more important is actually how much air we're moving through the alveoli. And so you can get an alveolar ventilation, V with a dot, sub A. And to do this, you're going to take the respiratory rate times the tidal volume minus that dead space, right? So I don't have enough room on here. Let me do it down here, right? Respiratory rate, let's keep that at 12. Times, oh my goodness, tidal, parentheses, tidal volume. I might have better luck just typing this. Minus the dead space, so volume of that dead space, which we said is about 150 mils. So if the tidal volume, right, remains 500 minus 150, that's going to be like 350. 350 times 12, now you're down to about 4.2 liters per minute. That would be your V sub A, right? The alveolar ventilation. Okay, there's a little bit of math that you might possibly have to do there, right? But it's not too bad. Okay. And you do not have to memorize all of these volumes. Don't worry about memorizing all of them. I would say you should know like tidal volume is about 500. You should know that a normal respiratory rate is 12. Other than that, if you need any of these volumes, I'll give them to you. Okay. Okay. One other note, right, is as we're looking at this graph, right, the normal tidal volume, in, out, we would call that quiet breathing, right, rather than forced breathing that you see in these maximum inspiration and expiration. And so in quiet breathing, right, or sometimes that's called diaphragmatic, in quiet breathing, you can either make it more diaphragmatic, right, you can be depressing the diaphragm, or you can breathe pretty shallowly and what we would call costal breathing, right? So you're using more of the rib cage to get the amount of volume change that you need. In both of those situations, it's going to be elastic recoil that gives you exhalation. So we would talk about the muscles involved in quiet breathing as the diaphragm and the external intercostals, right? When we start getting more forced in our breathing, that's when we start bringing in some of those accessory muscles. So things like, and they're showing you in here, the internal intercostals. Ooh, good review from AMP1. Internal intercostals, serratus anterior, pectoralis minor, transverse abdominis, your abdominal obliques, rectus abdominis, and on, right? So those are considered accessory muscles for inspiration. Expiration, again, in quiet breathing, it should be a passive elastic recoil. Active exhalation is going to require the internal intercostals and a lot of those abdominal muscles again. Okay. Okay, holy smokes. So that really just covered ventilation, right? The physical movement of air in and out of the alveoli. I will pick up the pace here. So the second thing that we said was part of external respiration then is gas diffusion, right? How do we get gas from the alveoli to the blood? So notice here we're still in this external respiration. The rules on how this is going to work, here's the picture from your book 2316. I like mine. The rules on how this is going to work, right, that govern the diffusion of gases. This is going to be ruled by the partial pressures of those gases, and then which we'll talk about Dalton's law there, right? The partial pressures, and then also how that gas moves in and out of a fluid, and that's governed by Henry's law. So a couple more laws. I know it's physics, but you can do this. So let's talk first about Dalton's law. I don't think I get to type in here. Okay. So Dalton's law, sometimes I call this the duh law, okay? Because what it says is, remember our atmosphere was that column of air sitting on top of you, right? And how heavy was it? 760 millimeters of mercury. Okay, so what is it made out of? What gases are in this column? It turns out that it's about, where are we? 79% of this column of gas, right? All of this is actually nitrogen. So 79% nitrogen. Okay, and then about 21% oxygen. And if you're doing math, you're going, well, that makes 100%. 21% oxygen. And then that's a little bit of rounding, right? We still have a little bit of water vapor, maybe like 0.5% for water vapor. And then about 0.04% CO2. So a very tiny amount. It's like this line, maybe. There we go. CO2, 0.04. So like 0.04% of a percent. Sorry, and these are all percentages. Is CO2. 21%, 79%. Okay, so all Dalton's law says, right, is that each gas contributes to the total pressure in accordance to its relative abundance, right? So basically what you would do is you would take 760 times 0.79, 79%, and you would find out how much of the column weight is nitrogen, right? I don't have that in front of me. I totally don't have that in front of me, right? 21% of 760, that I actually have in front of me. So the PO2, the partial pressure of oxygen, again, would be if you took 21% of 760, 760 times 0.21. Go ahead, like actually do it, work it out, right? And what you're going to get is 159. So now I could probably work this out, right? If you did 0.79 times 760, my guess is you're going to come up pretty darn close to the partial pressure of the atmosphere of nitrogen being 600, right? So that would be the PN2, the partial pressure of nitrogen, okay? You could calculate the PCO2. We're going to focus on oxygen for now, though, right? So again, if you took 0.04, well, that's percent, 0.0004 times 760, that would give you the PCO2. Okay, let's look at oxygen, though. Okay, so at sea level, right, when this whole column weighs 760, 21% of it's oxygen, so the partial pressure of oxygen is about 159 millimeters of mercury. So Dalton's law just really says, well, 159 plus 600 plus, plus these little other gases equals the total. Duh. Okay. The sum equals the total. So what we're interested in this for, though, is as we look at the partial pressure of oxygen, the percentage of the atmosphere that's oxygen doesn't change, right? It is always 21%. But if you go up in altitude, this whole column shrinks, right? So here I'm going to draw you over here, but you've climbed a mountain. So the top of the atmosphere is still in the same place, but the column of air on top of you, on top of the mountain, is shorter, right? So the overall atmospheric pressure decreases. And so now when you take, so say at Steamboat, right? Our atmospheric pressure here is actually usually right around 599 millimeters of mercury, right? Now, when you take the 21% of it that is oxygen, so 599 times 0.21, you're going to get 125. So the partial pressure of oxygen here in Steamboat, the PO2, I'm going to say 125, of 125 is less than at sea level. So to your body, there is less oxygen. It's the same percent of the atmosphere, but the partial pressure, the actual availability is less. Now, you can do the same thing, right? Like say you climb a big mountain, you climb a 14er, right? You have even less atmosphere above you. At the top of a 14er, right, a 14,000 foot mountain, the atmospheric pressure is around 460. So you can do that math, right? 460 times 0.21, you get about 96 millimeters of mercury of oxygen. And so that's why when you're climbing a mountain, right, or when you first moved to Steamboat or altitude, you probably noticed that it didn't feel like there was that much oxygen available. Okay, so that's Dalton's law. Duh. The sum of all the parts equals the total. The other half, right, of what's going to, what kind of rules the diffusion of these gases across the respiratory membrane has to do with Henry's law. And Henry's law basically is saying that at a given temperature, right, so you hold that constant, but at a given temperature, the amount of gas in a solution, right, the amount of gas that actually diffuses into solution is directly proportional to the partial pressure of that gas. Okay, each gas actually has a special solubility in liquid. So for example, CO2 is much more soluble in water than oxygen is, right? So we have 21% atmosphere, but if you tested the oxygen in a river, right, fish need oxygen to live, you would find that it's actually very low amounts of oxygen in that river because it just doesn't dissolve in there very well. Okay, so I think the thing to keep in mind, we don't get really into the math of Henry's law, but the thing to keep in mind with Henry's law is that basically the partial pressure makes a difference, right? It's almost like pushing that gas into a liquid. So for example, here we go. So I was having a little water refreshment here. As the atmosphere is bouncing off this water, some CO2, some oxygen are dissolving into the water, right? But not very much, right? If you want to carbonate something, had my LaCroix earlier, right? If you want to carbonate something, you have to up the partial pressure of CO2, right? And so they're infusing this liquid with carbon dioxide. But what happens as soon as you crack the top, you hear that psst, that's the CO2 trying to leave, right? Because it's no longer under pressure, okay? And so what we'll see is with gases is they want to move from areas of a higher partial pressure to lower partial pressure. So when we forced all the CO2 in here, high partial pressure of CO2, as soon as you crack it open, it wants to move away, right, out of the can. The same thing works in your blood vessels. So notice what's happening here. So we inhaled, right? This is the alveolus. And let's call the partial pressure of oxygen in the alveolus 100. Now, we saw at sea level that the partial pressure of the atmosphere, partial pressure of oxygen in the atmosphere should be about 159. But realize by the time you get down to the alveoli, you've been mixing, right? There's kind of dirty air still in the lung. There's all that dead space, etc. So it's not perfect. Okay, so let's call it a partial pressure of 100. Oxygen wants to move into the bloodstream because look at what the pulmonary arteries are bringing to the lung, right? This deoxygenated blood only has a partial pressure of 40. And so oxygen wants to go from high concentration to low concentration, right? Here, this is the rule. Everything's going from high to low. But the same thing happens, right? We were bringing CO2 back to the lungs, right, to get rid of it, and its partial pressure is 45. The reason it moves across this respiratory membrane is because the partial pressure in the alveoli is only 40, okay? And so it's going to move down its concentration gradient, okay? So the diffusion across, so we call this the respiratory membrane. Remember we talked about that in the first video. The endothelium of the alveolus, the endothelium of the capillary, the basement membranes, the interstitial space, right? Our respiratory membrane. It ends up being actually really efficient, okay? So the differences in partial pressures between the blood and the alveoli end up being substantial. The greater the distance, the difference in those partial pressures, the faster that diffusion is. It also works very efficiently because that distance is extremely short, right? We wanted a thin respiratory membrane. The gases that we're talking about, O2 and CO2, they're both lipid soluble. They don't actually need any kind of transport. They move directly, they diffuse directly through the membranes. They also diffuse through the surfactant. Remember that alveolus had like a little liquid and surfactant layer. They go straight through those, no problem. This is also really efficient because we have a huge surface area, right? We said it's like a tennis court. And then we also make sure to match the blood flow and the airflow. We coordinate those. So blood flow actually increases around alveoli that have a higher partial pressure of oxygen because we're trying to pick that up. Okay, so then what we need to look at is kind of what this arrow is showing us, right? The transport. Okay, so oxygen, give me 1 second. Okay, so our third and final step then of that external respiration is really how we're carrying, transporting these gases through the blood. So oxygen is carried by hemoglobin, right? Oxygen reversibly binds to hemoglobin. I have reversibly underlined, right? Because one of the things I want you to realize is that hemoglobin is actually this crazy protein, right? The heme group, the iron group on there, is willing to grab onto oxygen, right, and transport it, but it also lets go, right? It's not that hard to imagine a molecule that, say, grabs on to something else, but the fact that it reversibly binds, okay? So we want to pick up oxygen at the lung, grab onto it, but then we have to deliver it at the tissues, right? If it only does one of the two things, it's worthless to us. So it's actually a really cool molecule. And so when hemoglobin binds oxygen, we call it oxyhemoglobin. Sometimes you'll see it with like an HB for hemoglobin, HbO2, right? Oxyhemoglobin. And again, recall, if you look back at the blood chapter, that each of our red blood cells ends up carrying something like 1 billion molecules of oxygen. So we're moving a lot of this. All right, so here is the oxygen hemoglobin saturation curve. And this is, I'm sure we'll talk about this more in class. This one tends to be fairly confusing to people. But basically what we're measuring here, so notice over here on the y-axis, we are looking at our percent saturation. This is the percentage of all of our hemoglobin that is bound to oxygen, right? How much of it is saturated? How much of it has its 4 oxygen molecules bound? So what you'll see here is there's that PO2, partial pressure of oxygen, that we just saw in the previous picture, and the alveoli was around 100, right? And so as you would expect, right, at sea level, normal breathing conditions, almost all of our hemoglobin is 100% saturated, right? 97.5%. And so this would be if you put a O2 saturate, what is it? Pulse oximeter, pulse oximeter on someone's finger, it's measuring the oximeter part, is measuring that partial pressure of oxygen. So what we see here then is that areas with a lower partial pressure, say in those working tissues. So this is over at the lung, right? Say over at the tissues, right, where that partial pressure of oxygen is lower, notice we have a lower saturation, right? And so this makes a lot of sense. So when we think about the binding of oxygen to hemoglobin, the factors that are going to affect binding are things like the PO2 of the blood, right? So what this means is that the areas of lower oxygen, we're actually going to be delivering oxygen, and places of higher oxygen is where we are going to bind oxygen. So we have this pretty steep slope on the oxygen hemoglobin dissociation curve or saturation curve, either way. And so what this tells us is that pretty small changes, right? If I went from, what was I at, maybe 30 down to 20, I have a pretty steep slope here. So a small change in PO2 can lead to a big change in how that oxygen is either bound or released. And this also gives us kind of this automatic regulation of oxygen delivery. Places that have less oxygen get more oxygen. Okay. The other thing that's going to affect the binding are things like pH and temperature. And there's also something called 2,3-BPG. So let's actually... I thought your book actually did a nice job with this. So this is figure 2319, right? And again, here we're looking at the PO2. See, they laid this out. PO2 in the lungs. At the lungs, you should be almost fully saturated. Your bucket of oxygen should be almost full, right? When you're in these tissues where cellular respiration is taking place and they're using up oxygen, we have a lower saturation. We're delivering some of that oxygen to the tissues. And then if you exercise, and man, those quads are really burning as you're out on a run, right? As you're using up the oxygen, will deliver even more oxygen, right? So that's kind of that automatic control. Okay. So then this is the fun part of this curve. So what we can see here, what they're showing is what's called a right shift. So notice the red line is the normal, and this blue line is showing a shift in the curve to the right. And so these are going to be things, a right shift is what is going to help us to deliver oxygen. Right shift helps us deliver oxygen. You're just going to have to write that on your own. Okay, so notice it's things like a decrease in pH, as in the blood, the tissue being more acidic, like lactic acid tissue that's working hard, is going to have a lower pH. Okay, and that's calling for more delivery. Higher CO2, again, that's a sign of cellular respiration. It's calling for higher delivery of oxygen. And an increase in temperature, again, working tissue, it's calling for a delivery in oxygen. And this is what I really liked in your book, right? So notice what happens here. In that normal curve at whatever this PO2, the partial pressure of oxygen, say like 18, you might have been, what is that, 32% saturated. But as soon as we're exercising, you can have the same partial pressure, right, 18, but notice now how much more oxygen is getting delivered. They made that like 15% more oxygen being delivered, because hemoglobin changes its affinity for oxygen and it's willing to release. It's willing to deliver low pH, high CO2. Those two always go together, right? And then high temperature. And so this is actually super, super cool to me. The shift and the ability to release oxygen as a result of pH, that's referred to as the Bohr effect. So decreasing the pH, more oxygen is released. Okay. 2,3-DPG fits in here too. Oh, and now does your book call it BPG? Same thing, bis or dye. Anyway, this is in red blood cells, a product of glycolysis. So it's basically showing that we're running out of oxygen. And again, we deliver more oxygen. Okay, the curve can also shift to the left. This is a little annoying, I guess, that they switched colors, but here's here's our normal, okay? And now they're showing a shift to the left here. As pH increases, CO2 decreases, temperature decreases. The way I think about this, right, this one you're like, oh, this is what active tissue is like. Now just think about either resting tissue or what I like to think about is the lungs, right? The temperature would be slightly lower at the lungs. We've gotten rid of a ton of CO2. The CO2 is lower at the lungs. You're right, not right next to that working tissue. And so now we shift to the left. And this is going to make us more able to grab onto oxygen, right? To increase the uptake of oxygen in the lung, to increase our affinity. The other way that I often remember that left shift is this. So think of this line as the normal oxygen hemoglobin saturation curve in an adult. Fetal hemoglobin is actually slightly different, right? The protein that a fetus produces is slightly different, and it has a higher affinity for oxygen. It needs to be able to grab oxygen from mom, right, through the umbilical cord. So it's starting out with lower levels of oxygen, and so it needs to grab them harder. And that's what happens with a left shift to this oxygen hemoglobin dissociation curve. You're able to fill up even at those lower PO2 levels, right? So notice here, if we looked at just a partial pressure of 30, adult hemoglobin would be at like 60% saturated. Look at how good fetal hemoglobin is at grabbing on. That's over 80% saturated, okay? And that would be a left shift. Okay, so hopefully that makes some sense. This is typically one of the things that people find the most confusing. My encouragement is to think about a right shift as delivery. What does working tissue look like? And a left shift, maybe think about fetal hemoglobin. Okay, we're getting there. So that's oxygen transport. CO2 transport. Oh, here we go. Another picture. This one's CO2, I guess. Sorry. Okay, well, let's just look at this. So this is where we just were. Sorry, am I in your way? Down there. Okay, so here's oxygen from the alveoli, right? And it's moving into the bloodstream. Now notice, and I didn't talk about this, as oxygen is being carried, right? There's a teeny tiny bit that ends up being dissolved in the plasma, but just like in the fish example, oxygen does not dissolve well into water. And so most of it is going to be carried bound to hemoglobin, like we were just talking about. Okay, and so this is where we would picture at the lungs the hemoglobin picking up oxygen, and then they're just showing you, okay, we travel through the body and now we need to deliver oxygen to those peripheral tissues, to our cells. And so oxygen is released from the hemoglobin and it diffuses into the cells. And it diffuses into the cells because they have a lower partial pressure of oxygen than we saw here in the blood, right? So it's just diffusion. Okay, so that was oxygen. But now we need to look at how CO2 is carried, okay? Your first step is to think about why is CO2 coming out of these cells in your body? Like, where does the CO2 come from? And if you said cellular respiration, you would be correct, right? So remember, we're delivering oxygen so that your mitochondria can break down glucose and capture the energy. The byproduct is CO2. Okay, CO2 is a waste product. We need to get it out of the body, right? We want to exhale it in the lung. CO2 is carried really 3 different ways. So some of our CO2 is able to dissolve in the plasma, and that actually ends up being around 7%. It's actually much more soluble than oxygen is. So 7% of your CO2 is in the plasma. About 23% is carried by hemoglobin, right? So hemoglobin does have an affinity For carbon dioxide, it will reversibly bind carbon dioxide as well. We call that carbaminohemoglobin. And it doesn't bind to the heme group. It's actually binding to the protein portion, which I think is why they throw that amine. So carb, like carbon dioxide, amino, like amino acid, hemoglobin. Okay, so about 23% of your CO2 is carried that way. The majority, if you did some math, we got 70% left, is going to get carried in this whole crazy, like whatever's going on here. Okay, it's not as bad as it looks. You have seen this before, and you will see this again. So what's going to happen is the majority of our carbon dioxide is actually going to join together with water and form carbonic acid. So H2CO3 is carbonic acid. And notice again, H2C, and then we have 3 oxygens. So H2CO3, literally you put CO2 and H2O together. Okay, so you have an enzyme, carbonic anhydrase, that's in your red blood cells, and it runs this reaction. And then carbonic acid is going to dissociate. It falls apart into bicarbonate and hydrogen. Okay, the bicarbonate is, let's start here. The hydrogen, right? If you suddenly had a ton of hydrogen, that would do what to your pH? If you said lower your pH, that's correct. It would make you very acidic. And so that hydrogen is actually going to bind to hemoglobin. And in that way, hemoglobin acts as a buffer, right? It accepts those hydrogens and keeps the pH of the blood from shifting dramatically. The bicarbonate is going to diffuse out of the cell and actually just be carried in the plasma as bicarbonate. Okay. The only thing we do here is, since it has a negative charge, if it tried to leave the cell, eventually the loss of negative charges, like the electrical gradient, would keep more of the bicarbonate from diffusing out. And so we have what's called the chloride shift, and you see it swapping essentially for a chloride, just a negatively charged ion. So that's not a huge deal. That's just allowing more bicarbonate to leave. Okay. So then, keeping that in mind, when we get to the lung, all these steps need to reverse. And so this looks a little crazy, right? But bicarbonate is going to come back into the cell. It's going to pull hydrogen off of the hemoglobin, reform carbonic acid, and then carbonic acid breaks apart into CO2 and water. The water stays in the cell. The CO2 diffuses into the lung. And it diffuses into the lung because there is a lower partial pressure of carbon dioxide in the alveolus as there is in the blood. And again, we pull the carbon dioxide off our hemoglobin, and we pull the carbon dioxide out of the plasma. Okay, try to get rid of all that carbon dioxide. It is a waste product. Okay, so this image is a little crazy. Some people like to lay it out a little more linearly instead of this whole reaction kind of taking place in the top of a red blood cell. Totally get that. So in your book, this is more what it looks like. So they're showing the CO2 coming from the tissues. Here we see CO2 binding to hemoglobin, CO2 joining with water, becoming carbonic acid, splitting to bicarbonate and hydrogen bonding to hemoglobin, bicarbonate moving into the plasma. There's the chloride shift. Okay. One thing that is cool that they are pointing out on here is notice that when CO2 binds to the hemoglobin, it actually decreases the affinity to oxygen. So it's almost like kicking oxygen out to these working tissues. And that's true. So this is a little more linear, if that helps you. And then at the lung, of course, everything is reversing. Okay. We're almost there. Okay, let's just wrap this up real quick. So just like every other area of our body, what we're going to see as far as regulation goes is that we do start out, as far as regulation of respiration, that there's a local level, and then we'll talk about maybe what the the nervous system and the endocrine system can do here, though mostly in this case it's nervous system. So on the local level, right, tissue activity. So as those cells are actually doing cellular respiration, we're increasing the partial pressure of CO2, and we're seeing a decrease in oxygen, right? It's being used up at these tissues. So this increases the difference in partial pressures, right? More CO2 wants to move into red blood cells, more oxygen wants to come pouring out of red blood cells. So this is automatic. Hemoglobin sets us up for this, right? We'll also see in these areas where there's a lot of cellular activity going on, the increase in the partial pressure of CO2 actually causes relaxation in the smooth muscle. And so we get vasodilation. We get an increase in blood flow in response to increased CO2. At the alveolus, what we're going to see is that blood is going to get shunted towards the capillary beds, towards the alveoli that have more oxygen, right? Lower levels of oxygen actually help constrict those alveolar capillaries. And as more CO2 dumps into the alveoli, we get bronchodilation to actually dump this out into the atmosphere. So there's a lot of automatic things that happen, right? Autoregulation of respiration. We'll also see that the respiratory system runs, the nervous system runs respiration, particularly the medulla oblongata. Are you starting to sense that this is a really important region of the brain, right, in the brainstem? So there we are down in the medulla oblongata. And basically, and the pons plays a role here, kind of a secondary role. They're getting their sensory information from chemoreceptors, right? The carotid and aortic bodies, if you remember those. They're looking at the chemicals, things like oxygen, and they're measuring hydrogen. So they're looking at pH. Remember, pH is basically an indirect measurement of CO2. You also, the medulla has what's called the chemosensitive area, and it's actually it's testing basically the cerebral spinal fluid. And so it's looking for the partial pressure of CO2 and also the pH of the cerebral spinal fluid. And so the medulla can work to do things like change the pace of your breathing, right? If it notices that CO2 levels are rising, and it's typically CO2 is going to be the main stimulus here, it's going to tell you to breathe faster, right? To go ahead and get rid of that excess waste product, okay? That's a key point. CO2 is responsible for a lot of the regulation in breathing. Oxygen comes in a little more extreme circumstances, right? So high CO2 levels, that's referred to as hypercapnia, an increase in CO2. That's usually a result of hypoventilation, so not breathing enough. Hypocapnia would be low CO2 levels, and that could be the result of hyperventilation. So here's an example of how important that CO2 is as far as the regulation of breathing. So apparently high schoolers like to hyperventilate and then like hold each other against a wall or something. Anyway, if you hyperventilate, blow that air out, you can hold your breath longer because you lose the stimulus that says you should inhale and then exhale. You could try this, right? So if you want to run a little experiment, time yourself, hold your breath. And then before, I would actually make you do this in class, before you hold your breath the second time, hyperventilate. Blow off a bunch of CO2, hold your breath, you'll be able to hold it longer. Okay, because CO2 is that driver that tells the medulla to tell the diaphragm to contract, right? And to go ahead and breathe. You also get some information, so CO2 is the main driver. You'll also get some information from some baroreceptors. So a decrease in blood pressure will actually relay through the nervous system and tell you to increase the breathing rate. There is a stretch reflex that happens in the lungs. And then you also have some reflexes in regards to noxious stimuli. So when you sneeze or cough, like you're trying to propel an irritant out of the lungs, and there's a reflexive reaction that's set up by the brain in that regard as well. So you can do things like they're pointing out in this particular image that you can use, say, the cerebral cortex, higher level thinking, to actually tell yourself to hold your breath, right? But primarily, what we're setting up here is in the medulla, these rhythmic centers setting up a normal breathing rate. Okay, whew, that was an hour. I am done. I'll see you in class.